His present research focuses on nitric acid metabolism and the role in breast cancer biomarker development, and he's got a number of papers out, and one of the things we're particularly proud of, he's involved a number of students in his research. Students have presented at national meetings as well as been included in his publications. And today he's going to talk about his developing breast cancer marker that he's now seeking a patent on, and he's recently been in the news and all lately. Sir, they're all yours.
Thank you, Dan. Good afternoon. And it's again a pleasure for me to come up here and talk. I was here 3 years back, and Dr. Ling Kao brought me at that time, and I was talking about still nitric oxide metabolism and about arginine supplementation and the tolerance associated with it. It's good that I was able to continue the research for another 3 more years, and I moved it the exactly opposite direction now into breast cancer. So I'm still with the nitric oxide metabolism, but this time I would be talking about breast cancer. And as you all know, breast cancer is not going to be a homogeneous disease. It's going to be a heterogeneous disease, meaning it's going to have multiple subtypes associated with it. Among the different subtypes that are there with the breast cancer, when they did a large-scale gene profiling study, they found that there were 2 distinct categories of it, which was the estrogen negative and the estrogen positive subtype. Among them, for some reason, mine is not working. Okay, there you go. So between the 2, estrogen negative is going to be the more aggressive subtype, and it is the most difficult to treat, mainly because after the initial diagnosis, the prognosis of it is going to be difficult, especially for the first 5 years. And also, when we look into the survival rate of the patients who have been diagnosed with estrogen negative, we would see that compared to that of estrogen positive, an estrogen negative patient is going to have at least 2 times poor survival rate. And in addition to that, when we start to look into the different groups of people around us, if we look into African American, Jewish populations and other populations of people, we would see that compared to that of Caucasians, especially among African Americans, it's at least 2 to 3-fold more pronounced in African American population, and they seem to be testing positive for an estrogen negative type of breast cancer in an earlier stage of their life, especially when they are in their 20s and 25s. So that becomes a really important one for when it comes to the ethnic selectivity of this particular disease, because once someone comes with an advanced metastasis stage of this particular cancer, the survival rate of an ER negative breast cancer, especially when they are going to be a triple negative breast cancer, wherein they are going to have the negative or the mutant receptor for both the estrogen, progesterone, and the HER2, is going to be less than 5 years. So the survival rate of a person in an advanced metastasis with a triple negative is going to be less than 5 years, even with chemotherapy. So that becomes important criteria for them. So what are the treatments that are going to be available for these types of patients? Obviously, when we are talking about hormone receptor negative, hormone therapy is not going to be helpful for them, and any of the endocrine therapy also is not going to be helpful for these types of patients. Predominantly, what I was able to see, because I'm not a cancer biologist, my basis is on nitric oxide metabolism and cell biology, and I'm learning this as I'm going forward. So, talking with clinicians, I was told that some of the major therapies that are available are going to be chemotherapy, radiotherapy, and surgery are the options that these people, especially when it comes to triple negative breast cancer, that's what they are going to be exposed to. And how are they going to be diagnosed? That was my first question when I was trying to see and I was trying to talk with clinicians. And they told me that one of the ways that they can see it is by morphological imaging, such as ultrasound and mammograms, or by doing sequential biopsy of the patient once they have been diagnosed with estrogen-negative breast cancer, to follow up with that particular disease as to how it is going to progress and whether the therapy is working or not. But some of the major disadvantages associated with estrogen negative that they were able to point to me was that the cancer can grow between 2 scheduled mammograms. So even if it is going to be an annual screening that they have to do, within 2 months or 3 months between those scheduled mammograms, the cancer could grow. So that becomes a major challenge for us. And also, it's going to be asymptomatic. It's very hard to follow it until the cyst develops after a treatment or after a surgery has taken place. So between the surgery and the biopsy, we need to have something that would be able to monitor this particular disease as it progresses, or whether the therapy is working or not. That became the major criteria associated with it. And as we can think about biopsy or putting the patient to go through ultrasound or mammogram, it's going to be costly, and patient compliance is going to be one of the major factors associated with how much they are willing to go through this particular process. So this led to actually, when I went into the NCI website, I was able to see that this led to an increase in the requirement for biomarkers, especially non-invasive or less invasive type of biomarkers. And when I was looking into the cancer type and the type of biomarkers, the option of less invasive biomarkers were less. Most of them required the extraction of the tissue in order to see how it was working, and it did not have much of ethnic selectivity associated with it. Also, it was working for one group of people, but it was not working for another group of people. So it was not working in that particular perspective. And there was only 2 markers which was available through the blood, and those were the carcinoembryonic antigens and the CA15-3 and the CA27-29, but they would be helpful in detecting or saying the cancer reoccurrence in advanced metastasis. And speaking with the clinician, they said that if someone is going to be in their advanced metastasis, and going back to what I mentioned about in triple negative cases, the survival rate, even with chemotherapy, is going to be 5 years. So if they are going to be going through a terminal process, having a marker at that stage, how effective is it going to be, was the question that comes to our mind. So what is the crucial need for us? We need to have a biomarker that is able to catch this disease at an early stage, would help us or guide us through the disease progression process, would help us through the therapy outcome, whether the therapy is working or not, so that we are not over-treating a particular patient, and also have the ethnic distinction. Especially, I was more concentrated on the African American population, but since they were the more affected, 2 to 3 folds more affected compared to that of Caucasians or any other group. So that becomes an important criteria for them also. So before I jump into that, these were all the theoretical things that I was able to gather about it. And I was from a nitric oxide metabolism, I know that nitric oxide is a double-edged sword, meaning that for a certain type of cancer, an increase in nitric oxide would be beneficial in curing the cancer. And in other cases, an increase in nitric oxide would lead to poor survival. So I wanted, and also nitric oxide, there are 3 different ways by which nitric oxide could be generated. The nitric oxide synthase is the enzyme, but there are 3 different isomers of the same enzyme, NOS1, NOS2, and NOS3. And NOS2, which is the inflammation or the inflammation-inducing type of nitric oxide synthase, is associated with inflammation, and inflammation is the starting point for cancer. So this was my thought process as to how nitric oxide in general is going to control the cancer in general. And I wanted to know whether nitric oxide has either a deleterious effect or a beneficial effect when it comes to estrogen-negative breast cancer. So when I was thinking about it, I came across a manuscript from NCI, published one, wherein they said that NOS2 seemed to be a feed-forward mechanism for estrogen-negative breast cancer. So that was my starting point. So they said that, oh, NOS2 is working for us, which is good. And they also said that aberrant changes in NOS2 is seen with multiple solid tumors, such as colon cancer, prostate cancer, and in melanoma. So NOS2 seem to have the dual role in estrogen negative, wherein an increase in NOS2 or an increase in nitric oxide is going to correlate with poor survival for an estrogen negative breast cancer. And it's the other way around for estrogen positive breast cancer, wherein an increase in nitric oxide or an increase in NOS2 is going to be helping in curing that cancer. So that was, in fact, the starting point for me. But when we look into nitric oxide, we know that, again, I have to go and extract the tissue of the cancer in order to see whether it is increasing or decreasing, and it's not going to be beneficial for me. And since aberrant changes is occurring, and we have 3 different types of NOSes also, I wanted to say what is common between all the NOS metabolism and what could I correlate it with. When I was thinking about that, that's when NOHA, which I call it, which is basically an N-hydroxyl L-arginine, which is an intermediate of the NOS pathway. So I've simplified the NOS pathway. Basically, the NOS pathway requires your arginine to come into through the carrier mechanism, and the nitric oxide synthase utilizes the arginine to form NOHA, which is the intermediate, and the intermediate gets converted to by the NOS to nitric oxide. This is a simple, straight, one-way mechanism by which nitric oxide has been generated in this particular process. But the interest came in the sense that of the 3 NOSes, NOS1, NOS2, and NOS3, NOS2 is the calcium-independent mechanism. NOS1 and NOS3, which would be activated during a regular inflammation process, is going to be a calcium-dependent mechanism. And between these 3 NOSes, only the NOS2 is capable of effluxing NOHA outside. So, was that clear? So only NOS2, because it is a calcium-independent mechanism, is capable of effluxing the NOHA outside, which was my, oh, that's good. I have something which gets effluxed that is specific for cancer, that is specific for an inflammation-induced cancer, that would be, I can measure outside. But the next question is, well, it can go out, and if the body can reabsorb it from the systemic circulation into the tumor or into the tissue, I'm looking at nothing. So that's when I went into the KM and looking into the absorption mechanism. Apparently, the reason why I have the high arginine and a low NOHA here is arginine is produced in a body at millimolar concentration, while this NOHA is getting itself effluxed only in nanomolar concentration. So it's almost like you guys have $1 million and I have $1, and we are sitting in the same blackjack table. I wouldn't be allowed to sit in the same blackjack table, but there is a competitive inhibition happening there, which means that the NOHA is not going to go into the system until or unless the arginine concentration comes down dramatically, which is not going to happen. And even hypothetically, we think that, okay, the NOHA goes inside. But when we look into the Km of the NOS2, the Km of NOS2 for NOHA is at least 2 times higher than that of arginine, which means that NOS2 is going to favor more of conversion of L-arginine to nitric oxide rather than trying to use NOHA for the production of nitric oxide. And the irony is, it goes through the NOHA for the production of nitric oxide, but it still favors arginine for the production of nitric oxide, and any NOHA that in case if it comes in also, it's not going to use it up. So that was something that I had, and that's what I have mentioned here also, wherein it is a very strict association with NOS2, and the reabsorption metabolism is close to almost impossible until or unless it can happen in a very dramatic way. So this was my theory to see whether NOHA could be measured outside, because now it makes sense that it gets itself effluxed and it stays outside. I don't know the pK of how it is going to get itself eliminated, and that is my next step to see how it is going to get eliminated, what is its rate of elimination and all those things. But all I know is it stays outside and it doesn't get itself reabsorbed. So in order to first test it, obviously, I go to my cells, and I started with Caucasian cell line to first start it because that's what I had initially. And when I was testing with a couple of Caucasian cell line with estrogen negative and estrogen positive, and I first started with monolayers, and I went to a boot camp in NIH-sponsored boot camp in Maryland, and they said monolayers are ancient. You got to go something beyond monolayers in order to try to understand what it does and to have a much better translation between an in vitro to in vivo. Anything can be seen in a monolayer. So what I did is I developed a 3D spheroid. And we have an incubator facility in Brunswick, wherein Inspiro is the company which actually has that apparatus, or it's basically a 96 well plate, which is a V-shaped 96 well plate which has a hole at the bottom. So basically you put the cells into it and it grows as a spheroid. And the good thing about it is it will grow for up to 10 weeks. So it's not only that you have to subculture it every week, you can have the same cells growing for up to 10 weeks if you want. And since I was interested only in the medium, I can keep changing the medium from top, and there would be a tray. I can collect it at the bottom, and I can analyze it for 10 weeks if I want, and keeping the tumor going for up to 10 weeks. But the initial thing that I showed here after the boot camp, that's where I wanted to see, they wanted to make sure that I'm seeing it in multiple cells. So I went ahead and looked into 3 different ER negative Caucasian versus African American and 2 different ER positive Caucasian versus African American, and saw that there was a steady increase with a steady decrease in the NOHA with ER negative, and it was also specific for that particular Caucasian versus African American population. There was a distinction in the Caucasian versus African American population in the level of reduction in NOHA and an increase in nitric oxide synthase 2, as well as in the nitrite amount. And I measured nitrite mainly because nitric oxide has a half-life of few minutes or few seconds, and I cannot measure it. So I'm quantifying nitrite as a stable end product of the nitric oxide metabolism. So this is good. This is in cells. The next thing is I want to see whether it would be translated from the 3D spheroids from an in vitro model into a clinical study. That's what I did. And I picked 5 different ethnic groups. I picked African Americans, Caucasians, Hispanics, Asians, and Jewish populations. And I went to CHTN. I was being directed instead of me going to CHTN. I was being introduced by one of my colleagues to go and look into CHTN, which is a cooperative network operated by NCI or funded by NCI, so that they would go, it's not a biobank by themselves, but they coordinated with all other biobanks in the country. So if anyone anywhere has an African American low grade stage 1, that was my criteria that I was having for the de-identified samples, has that, they would ship me the plasma that I want. So that helps me. It takes time. It takes about 6 to 8 months for me to populate everything, because sitting in Maine, I cannot get the other ethnic population at this particular number. So that's where I was very grateful that CHTN, Bioconserve, and Tissue for Research, I have to go through them in order to get all these samples from them. And it also helps me in understanding the diversity, because this is not sample coming concentrated from Maine or in the New England area. This is coming all over from the country, maintaining the same low grade stage 1 breast cancer. And I can see a distinct correlation with ER negative, wherein there is a drop in the NOHA level compared to that of ER positive or control groups. And between the ER negative groups also, the African American have a distinct left shifting compared to that of Caucasians, Hispanic or Asian, and Jewish population, because it's n equal to 5, was in between those two. Was it clear? So this is good news for me in the sense that I was able to see something happening in an earlier stage of cancer, of the ER negative cancer happening, and I can see it in the blood, which means that a needle prick amount is all I would need in order to run it. And any of the sample assays that you are seeing here or the results that you are seeing here are validated by 2 different methods. I validated it initially by LCMS, and I also validated it based upon a pilot ELISA assay that I was able to do. And I would like to thank Ed and Nick for this, because they funded a portion of it, and the College of Pharmacy who funded another portion of it in order to make the custom antibody creation. And it's from Thermo Fisher Pierce, and it was a proof of concept, and they helped me in making the monoclonal antibody raised against NOHA, so I can actually run the assay for that. So the main purpose of it is, in the long term, I can move more towards the ELISA assay, because that would help from the diagnostic purposes, making it more of point of care testing. That's the hope. So when I'm doing something like that, I wanted to have an ELISA assay to go with it. And it also helps me in validating by 2 different methods. I'm validating the method in one way and also validating the assay, validating my results, whether it is cross-assay compatible also. So it has been validated by 2 different methods. So one good thing is I'm able to measure NOHA, but for it to be measured in a better way, the next thing I have to know is whether it's going to be stable or not, because once it comes out and I'm able to extract the plasma, by the time it gets shipped, or even if I'm going to assay 2 hours after versus now, if it is not going to be stable, then I don't know whether the reduction that I'm seeing is a true reduction or it's going to be a false positive or a false negative that I'm seeing here. So the one that on your left is basically what I did with the 3D spheroids. Again, the advantage of the 3D spheroids is I can grow it up to 10 weeks. So I was trying to monitor what happens. Oh, I'm sorry. I misspoke here. What I did here is I spiked the medium sample with NOHA, and I was monitoring it, how it was reducing, or whether there was change in the stability level. And I found that at higher temperature, this was the major challenge, there was a drop after 2 weeks. And more importantly, when I checked it in the plasma, 4 degrees room temperature and -80, there was stability that was happening. But anything above that, it was not stable. Initially, when I looked into this, I was happy because I was having stability for up to 7 days. But as I move forward with this presentation, I have much better data coming up, so this is ancient for me now. And the next thing that I have in this process is I wanted to see whether NOHA is able to be predicted based upon disease progression. Again, I went to my 3D spheroids and I was growing them over 10 weeks, and when I took those spheroids, and Inspiro has this imager wherein they would be able to see it and see whether there is any difference in the way it looks from that of a regular cell. So when I was looking into it, week 3 cells were looking, cancer cells were looking closer to that of a regular cell line, but or a normal cells, but week 6 they were showing to be more different from that of a normal cell line or a healthy cell line. So that was my initial process of thinking of whether it could be like a low grade versus an intermediate grade. So I was playing about with it and tried to see whether there would be any difference between a week 3 versus a week 6 NOHA level in that. And there was a difference in the week 3 versus week 6 NOHA level. There is a progressive decrease that was happening there. So again, this is in vitro data. How does it correlate it with the in vivo data? And before I wanted to start this one, again, one of the clinicians helped me in trying to see from a clinician perspective. They were looking at the low risk versus high risk patient. Oncotype DX for estrogen positive, they have the genetic profile of predicting the reoccurrence or the risk of estrogen positive tumor happening there versus mama print, which is for both estrogen negative and estrogen positive. I'm just saying these things because I learned it just now through this process. So if you ask me what is mama print and how does it going to differ from that of Oncotype DX, he might be able to say something from the genetic perspective, but all I know is they are able to provide a score, and a lower score means low risk, and a higher score coming out from that means that they are highly susceptible to get any one of these particular diseases. So that's what was told to me. So based upon that, again, I went to CHTN and asked them, well, I'm trying to test this. Could you help me in any of your de-identified samples with any of them having these types of criteria that you can mention it to me or provide me the samples with? They were able to provide me grade 1 and grade 2, and this is a very small population, n equal to 3, because if I have to do it at a bigger scale, I have to they have to set up a protocol and they have to go through the process. So I did it at a very small scale. So it's grade 1 as a proof of concept. So it's grade 1 versus grade 2, low grade versus intermediate grade, and low risk versus high risk. And the interesting thing is there is a disease progression correlation, and there is an ethnic specific correlation also happening, which was good for me. So these things help me to understand that now I can make a NOHA assay, either by an ELISA assay or an LCMS assay, taking just a needle prick amount of the blood at a cost that would be similar to that of a cholesterol diagnosis with a turnaround time of a cholesterol diagnosis, and I can detect it earlier than other blood tests or other blood-based marker that is available, and with ethnic specificity in which I can. And now this has to be a surveillance type of long-term longitudinal outcome measurement that I have to do, but at least we could head in the direction of saying that genetic risk assessment could be correlated with that of the NOHA measurement that I could see. So, but this one would require a large-scale study for me to better correlate or even validate, saying that, yeah, I can correlate it, because for me, n equal to 3 was not enough. But before I go and do the progression study, again, I go back to my 3D spheroids and try to see how does it behave, because I want to see whether it is going to go show me a trend, and surprisingly, it does show me a trend of a reduction in NOHA in the medium and a reduction in NOHA within the tissue itself or within the tumor itself, with an increase in NOS2 and an increase in nitric oxide, both of which are going to show poor survival for us, and these happening only in ER negative and with ethnic distinction happening for me. So this gives me some confidence for me to go and plan how I want to do my longitudinal outcome study when it comes to clinical patient. And it also showed me one additional thing. Compared to measuring it in the tissue itself, so if I compare to taking a biopsy and taking it from the patient itself, the sensitivity of measuring the NOHA in the medium was better. So it is much better to do it in the medium, because it took me 3 weeks here under the in vitro condition for it to be shown, whereas within a week I was able to see the difference in the medium. So And the other thing that I wanted to see or cross-check myself as to I'm looking into the right one and not a false positive is NOHA is known to be a good inhibitor of the arginase metabolism. And when arginine comes in, it can go through the nitric oxide synthase metabolism to produce nitric oxide, or it goes to the urea cycle, wherein it goes through the arginase and it gets itself refunded back. But since the arginase is getting inhibited, I wanted to see how the arginase activity is going to be correlated here. So if there is going to be less NOHA inside, if it's going to get itself shunted out, the arginase has to go up, and that's what was happening. So just to confirm that the arginase that was present was getting itself shunted out and it was not inside the cell, that's what I wanted to see here. And this is where I'm getting excited about it, because so far I was looking at regular needle prick amount of blood and getting the plasma out of it to measure it, and I was talking about having its stability for about 7 days. But in order to make it more user-friendly, so if a person is in rural Maine and they don't have access to these types of medication and these types of facilities that we have in MMCRI or other places, then we have to be able to ship the sample easily. And one of the major drawbacks is shipping it at higher temperature, room temperature, or say 42 degrees, and ease of shipping without putting it in ice and things like that. So the freeze thaw, I wanted to avoid it. So how would it work in dried blood or dried plasma? That's what was the thought process, and it seems to have a good stability. So this is a commercially available sort of a business card like looking strip, and this is from Shimadzu. Basically it's a glorified 0.22 micron filter. That's what I see this. So you put the blood about 25 microliters on top, and it has a micron or a filter that goes through it, so it separates the cells from the plasma, and they say that 2.5 microliters of the plasma gets onto the disk, plus or minus 0.1 microliter. So that gives us a defined volume for us to even go and look into it, what we have. And this one is from only LCMS, from the ELISA. From the LCMS, we have a 72-hour data so far, and it looks promising enough. But from just the ELISA assay, we could say that it has a stability at 42 degrees for up to 14 days. So, which is good. So anyone who wants it to have it can take a needle prick amount of the blood on the strip. And the good thing about that strip is the blood flows through a small amount and it becomes red, so they know that how much of blood they have to put it in. So they have the quality control associated with it as to how much makes 25 microliters for them to do that. So they can just ship the sample to us and we can extract it and see whether they are in track of it. So even when we are setting the longitudinal outcome study, it becomes easier for us to collect the samples this way rather than collecting it via plasma and trying to, because each time I got the plasma from CHT and I measured it at the time I got it, and also at the time when I have all the N number with it, just to make sure that they are the same and the freeze thaw effect does not have an impact on it. So where are we moving forward? As I said, one of the things that I wanted to see is the disease progression that is going to be associated with it and how it is going to be correlated. So we can look predominantly at the BRCA1 and BRCA2. Whoever gets themselves into the trial, say we can work initially with MMCRI and then later on with Central and Eastern Maine, and try to see how they are going to correlate with it. And once they are enrolled, before surgery, how the NOHA level is, after surgery, how the NOHA level, and we can have a baseline before all of this to see whether the trend goes up or goes down, and we get an idea of how we can trace it, and it is much better than the sequential biopsy that would be there. and we can catch it much earlier than the sequential biopsy method that could be there. And obviously, by doing that, we can have a more personalized medicine or therapy and avoid overtreatment. That is another thing. And by doing the progression study in the long run, probably, and if I can link it with the genetic testing and all those things, when it comes to rural Maine and people who don't have access to it, it can be made as an early detection method also. So it could be a primary mode of screening in the long run. So those are the thought process that I have, and that's all I have. Questions? Yeah.
So when you got the samples from the patients, were these very late stage cancers, or were they earlier, or did you do a range? Because the amount of cancer or the number of mets might have influenced the levels.
These were patients who have not gone through any treatment. These were early stage, low grade. They were at the time of diagnosis. They were at low grade, stage 1, without any treatment. So I don't know how it is going to behave with and without therapy. That's one thing that I wanted to measure with chemotherapy, or how it is going to respond to Taxol. So that's one thing that I wanted to see, because if it's going to have any drug-based interaction associated with it, then it's...
Right.
Yeah. Then.
So relate to your earlier stage detection. So I just wonder, I'm not familiar with the biochemical pathways. So this, for normal people who do not have breast cancer or any type of cancer, is NOHA as negative?
NOHA would be high.
Very high.
So if it drops, it's almost like a FICO score or your credit score. The more it drops, the worse it gets.
So then actually as the disease progresses, you actually will get to a point maybe not detectable at some point, right?
Yeah.
One of your features has
Maybe at a very later stage, there could be a possibility that I may not be able to see the NOHA at all.
And have you tested other cancer cells just to see how specific the assay is for cancer?
I didn't check other, you mean other solid cancers like colon. I didn't check it because I don't have access to it, and I don't have access to it. Yeah.
I'm sorry if you've already mentioned this, but have you tested this on breast cancer in males?
Yeah, that's a good question. I have not tested it on breast cancer males. So that is another population that I have to check. So far, I've concentrated only on the female patient population. Yes, Tom.
So any speculation why the vast difference in African Americans?
One speculation, and please correct me if I'm wrong, is eating habits or the environment, or how they are, if they are of a lower income category, or if they're camping more on McDonald's type of a food. And not that I'm against McDonald's, but it could have an impact, because one of the manuscript, they were looking into that, and they showed that it is more prevalent, negative chances of having it is more on a lower income category than on a higher income category, and that could be it. But
No, I mean the NOAA. Why would that be so different between?
I don't know. Disease-wise, I was able to do it, but this I couldn't know.
But were they different in the controls?
No.
More specifically different in the tumor therapy,
Tumor.
right?
They are more.
Or their response to the tumor and the growth in their body than it was at a baseline. So it's not a predictor, more as a response.
One of possibility is, since it goes through the carrier system, maybe the number of carrier, Y plus carrier, the cationic transporter mechanism is more expressed in an African American compared to that of a Caucasian, and that.
African Americans, any idea how diverse? Because that's a broad category, and there's a huge difference between North Africans and Sudanese, or at this point, they're just called African Americans. Okay.
That's what.
Yeah, no, I understand.
And even when I was presenting this Jewish one, they said that there are 5 categories of Jewish population, and I didn't know which category it was. So, any other questions? Or any thought process or something that I could look into it? For example, the male breast cancer that I haven't thought about, I can look into it. But
All right, well, thank you.
thank you.